Hello, welcome again to Kim's Bioproduction Academy. Today, we want to discuss about cell division. And on this cell division, we'll have to, that is either by mitosis or meiosis. And the first one, let's discuss first about the mitosis. This one will just be having one stage, circle. On mitosis, it will be undergoing some 5 stage or phases. And the first one, in the formula, we have something called IPMAT. On this IPMAT, that is I for interphase, P for prophase, M for metaphase, A for anaphase, and T for telophase. Let's discuss about each stage. The stage 1, that is interphase. These are preparatory stage. And on this preparatory stage, what is being entailed? The first thing, we have 3 functions there. Duplication of the genetic material. When we speak about the duplication of the genetic material, that's like we're speaking about replication. Second, we have synthesis of the new cell organelles. That is formation of new cell organelles. Which are those cell organelles? There are many. Mitochondria, Golgi bodies, centrioles. Third, we have synthesis or formation of energy. That is a replication. No, that is the synthesis of energy. That is formation of energy in form of ATP. That's its storage form. Again, at that stage, we have some features for that stage. There will be presence of nucleolus. 1. Again, presence of a nuclear membrane. So the nuclear material will be enclosed by a nuclear membrane. We will have, that is formation of the centrioles and even formation of spindle fibers. Sorry, we don't have formation of spindle fibers. That's the next stage. From that point now, the nuclear materials within that stage, we are calling it the chromatin. They are thread-like. They are not much visible. They are just thread-like. They are not yet chromosomes. They are thread-like chromatins. That is the end of interphase stage. Let's go to the second stage. We have said it's which one? Prophase. In prophase now, those chromatin materials, they'll condense and become more visible. Now they're forming chromosomes. They're being seen. They are becoming more visible. Second, those formed spindle fibers separate and move to the opposite poles. Once in the opposite poles, we get another point, which now leads to the formation of those spindle fibers. Spindle fibers start to form. Third, the nuclear membrane starts to break. It will not completely disappear, but starts to break. Nucleolus is the one that disappears. From that stage, what happens? We go to metaphase. At the metaphase, the chromosomes align at the equator. The chromosomes align at the equator. Those formed spindle fibers will lengthen to attach the sister chromatids at the centromere. They'll lengthens and attach the sister chromatids at the centromere. The nuclear membrane breaks completely and disappears. We go to Anaphase. Anaphase, at this point, the sister chromatid separates. And they will do what? They will move to the opposite poles, and this is due to the shortening of those spindle fibers. So the shortening of the spindle fiber will be pulling those sister chromatids towards the pole. That is the end of that phase. The last phase now is the telophase. The telophase in plants and in animals will be different. In plants, because of the presence of a cell wall, it's very rigid. You know that cell wall is very rigid. So there will be no constriction of the cell wall. So there will be formation of something called a cell plate. This cell plate forms to separate the cytoplasm. But before that, there will be something called cytokinesis. There will be separation or the division of the nucleus. And each nucleus, one will move to the parent cell and one will remain on the other cell. So it means we will have now two daughter cells being formed at the end of this stage. That is in a plant cell. In animal cell, there will be constriction of the cell membrane. Constriction, that is invagination of the cell membrane. Then again, after that now, the cytoplasm will be separated and there will be reformation. The division of the nucleus, that is through the cytokinesis, we form two nucleus, one for this other cell, then the other for the daughter cells, and one remain on the parent cell. Then from that point now, there will be reformation of some structures, like the nucleolus and the nuclear membrane. And that is the end of mitosis, where we form two daughter cells. Thank you. As we learn more, so our next slide will be for meiosis. Thank you.